Hello students, in previous lecture we have discussed factors affecting enzyme activity. Today we will discuss about specificity of enzyme and mechanism of enzyme action. So the learning objectives are enzyme specificity. There is the three types: specificity of enzymes, a reaction specificity, and substrate specificity. And second one object, it is the mechanism of enzyme action, lock and key model, induced-fit theory. So first of all, specificity of enzymes. Enzymes are highly specific in their action when compared with the chemical catalyst. The occurrence of thousands of enzymes in biological system might be due to specific nature of enzymes. The types of enzyme specificity: stereospecificity, reaction specificity, and substrate specificity. See, in stereospecificity, stereoisomers are the compounds which have same molecular formula but different in their structural configuration. Enzymes acts only on one isomer and therefore exhibit stereospecificity. For example, L-amino acid oxidase and D-amino acid oxidase acts on levo and destro amino acids respectively, or hexokinase acts on D-glucose, glucokinase acts on D-glucose molecules. In class of the enzymes belong to isomerase do not exhibit stereospecificity since they are specialized in interconversion of isomers. Second, it is the reaction specificity. The same substrate can undergo in different types of reaction, each catalyzed by separate enzymes, and this is referred to as reaction specificity. For example, in urea cycle metabolism, the amino acids molecules they undergoes the three types of the reaction: transamination, deamination, and decarboxylation. Here the substrate amino acids, but the substrate with the help of the enzymes, there is the formation of the three kinds of these reactions. Amino acid in oxidase stage, there is the removal of the amino group, there is the formation of the keto acid. Amino acid substrate with the help of the decarboxylation, means removal of the CO2 group, formation of the amines, means it is the reaction specificity. Next is the substrate specificity. Substrate specificity, various forms of the enzymes to enzymes. It may either absolute, relative, or the broad. Absolute substrate specificity: certain enzymes act only on one substrate. For example, glucokinase acts on glucose to give the glucose 6-phosphate. Means it is the depending upon the glucose is converted into glucose 6-phosphate. Enzymes, it is the glucokinase. It is the specificity. Means absolute that reaction Means in glycolysis pathway, glucose is converted into glucose 6 phosphate. Suppose this reaction involves into the in muscles, means absence of the oxygen molecules, there is the need of the enzymes. It is the hexokinase, or the same reaction involves into the liver. There is the need of the enzymes. It is the glucokinase. It is the specificity, substrate specificity. Second is the relative substrate specificity. Some enzymes acts on structurally related substance. Means maybe depends on the specific group or the bond present. For example, in case of the glycosidates, acting on glycosidic bond of carbohydrate, lipase cleavage the ester bond of the lipids, it is the relative specificity. And third, it is the broad specificity. Some enzymes acts on closely related substance, which is commonly known as broad substrate specificity. For example, hexokinase acts on glucose, fructose, mannose and glucosamine and not on galactose. It is possible that some structural similarity among the first four compounds makes them a common substrate for the enzyme hexokinase. So this is the enzyme specificity. Next, towards the mechanism of enzyme action. Catalysis is the prime function of the enzymes, no doubt. The nature of the catalysis takes place in biological system is similar to that of non-biological catalysis. For any chemical reaction to occur, the reactants have to be in activated state or transition state. Binding of the substrate to the active site of the enzymes causes bonding rearrangements that leads to intermediate state called as the transition state. The you see in diagram, there is the formation of the transition state and intermediate this catalysis stage or the uncatalyzed stage. There is the formation of the active forms of the substrate immediately preceding the formation of the product. As an enzyme speeds a reaction by the lowering the activation energy, less energy is needed to convert the reactant to products. Activation of the free energy. The enzymes required by the reactants to undergo the reaction is known as the activation energy. The energy required to convert the substrate from ground state to the transition state. Substrate needs a large amount of energy to reach the transition state, which then decays into the product. The enzyme stabilized by transition state, reducing the energy needed to form the products. The enzymes do not alter the equilibrium constant, they only enhance the velocity of the reaction. The role of the catalysis of the enzymes, comparable with the tunnel made in a mountain to reduce the barriers as illustrated, see in diagram. The enzymes lowers the energy barrier. Reactants, they are making the reaction go faster. Second, mechanism of enzyme actions. Let's see how to the formation of the enzyme substrate complex. The prime request to enzyme catalysis is that the substrate must combines with the enzymes at the active site to form enzyme substrate complex, which is ultimately results in the product formation. See in diagram first, enzyme active sites, substrate molecules binds the formation of the enzyme substrate complex. In catalytic activity of the enzymes molecules, see in diagram 2, enzymes in catalytic site of the active sites bind the substrate, the formation of the enzyme substrate complex. If the substrate molecules is converts into the product in form, the product it is leaving from the enzymes active site, the enzymes molecules they are the reusable. According to this process, the few theories have been put forth to explain the mechanism of enzyme substrate complex formation. The first hypothesis, it is the lock and key model, and second it is the induced fit model. Lock and key model, this theory was proposed by German biochemist Emil Fischer. This is in fact a very first model proposed to explain an enzyme catalyzed reaction. According to this model, the structural or conformation of the enzyme is rigid. The substrate fits to the binding sites or the active site just as key fit into the proper lock or hand into the proper glove. Thus, the active site of the enzyme is rigid, pre-shaped template where only specific substrate can bind. This model does not give any scope for the flexible nature of the enzymes. Hence, the model totally fails to explain many facts of enzymatic reaction. And the most important, bring the effect of allosteric modulators. See in the diagram, the enzyme's active sites, key it is the substrate, the enzymes, the key insert to the enzyme's active sites. But in this process, the enzyme active site it is the rigid, so that's why there is a need to the specific site of the substrate. And if there is the specific type of the substrate, then formation of the enzyme substrate complex. Otherwise, there is not the enzyme's active site is the selfly molded or not the formation of the proper enzyme substrate complex. In second form, there is the induced fit model, which is also known as the Coastland model. The Coastland in 1958 proposed a more acceptable and realistic model of the enzyme substrate complex formation. As per this model, the active site is not rigid and pre-shaped. The essential factors of the substrate binding sites are present at the nascent active site. The interaction of the substrate with the enzymes induce a fit or conformation change in the enzyme, resulting in the formation of a strong substrate binding site. Further, due to induced fit, the appropriate amino acids of the enzymes are repositioned to form the active site and bring about the catalysis. See in diagram, there is the substrate, enzymes molecule, the enzymes possesses the active sites, that active site, addition of the substrate, the enzymes selfly mold or the selfly change the conformation and they can occupy the substrate. Means it is depending upon the substrate, not the enzymes active site. Enzyme active site is not the rigid, it is the flexible in form, change the conformational form. So that's why substrate molecule they are easily occupied to the enzymes active sites. The formation of the enzyme substrate complex. The substrate gets converts into the product in form the product leaving the active site of the enzymes, and enzymes molecules, they are the reusable. So this is the induced fit model. There is the mechanism of enzyme action. Thank you.